So welcome everybody. Welcome to this webinar on hepatitis B research for the community. We really want at ICE-HBV to involve people who live with hepatitis B in our work, and that's why we're holding this series of webinar, and this one is focused on Europe, as you will see with our speakers. And we'll also be describing today not only cure research that's being done around the world, but also specific projects happening in Europe. But we also want to look at specific issues regarding hepatitis B in Europe, such as HBV and migrants. So this is a webinar format, so you can ask questions via the chat or the Q&As, and there will be a discussion time at the end for answering your questions. The program is on the slide right now. I will just give you a few housekeeping information. So right now, only the moderator and the panelists will be able to turn that video and no mix. We were building with ICE-HBV FAQ on our website on HBV Cure, and this is based on your questions. So please add questions on the chat, and even if we can't answer them today because we run out of time or we don't have the answer for you, we'll use these questions to build these FAQs next year. So you can also send your questions on cure research, hepatitis B cure research to info@icehbv.org. That we'll be very grateful that you tell us which information you need. And this meeting is recorded, and we're hoping that the recording and the slide will be shared with participants and will be on our website. Of course, that depends on the consent, but I think we'll have everybody's consent for today. And I think that's it for me. I just want to say, as we start, thank you to our sponsors, so ICE-HBV key industry sponsors. Without them, this wouldn't be possible. So they're Jensen, Antios, and Aligos, and also to our generous donors, the NRS and the Hepatitis B Foundation. With that, I think I can hand over to George and we can get started.
Well, thank you very much, Capucine, for organizing this very interesting meeting. My name is George Kalamitsis. I come from Athens, Greece, and I represent today the Liver Patient International, an international patient association based in Europe, but represents globally patients around the world that live with liver diseases. It's, I think, an excellent opportunity today to talk about the disease, hepatitis B research. A disease that affects millions of people worldwide, and many of the patients that are waiting for developments in current treatments in order to affect in a better way their life in the future. Since we do not have too much time, and I need to introduce to our first speaker, which is Dr. Fabien Zoulim, which is a medical director of hepatology department at the Hospices Civil de Lyon, and scientific director of the Department of Immunology and Virology of INSERM, Unit 1052, where he is leading the team of antiviral therapy on viral hepatitis. Professor Zoulim will have a speech regarding hepatitis B cure research 101. Mr. Zoulim, the floor is yours.
Okay, thank you, George, for the introduction. So now I'm sharing the slides. I hope it's all good on the sharing. So today I will try to give you an introduction on HBV cure research, and you will have other presentation later on specific topics regarding HBV cure, and hopefully the discussion will be interactive. So regarding HBV cure, I mean the first question is why do we need it? And as George was saying, I mean, we have more than 250 million chronic carriers worldwide, and very few are currently treated, and the drugs that we have big limitations. So they rarely cure patients, and sometimes they don't stop the disease in some of the patients, despite a good efficacy on the virus. So the main ones that are used currently are nucleoside analogs, with 1 pill a day, but these drugs do not eliminate the virus from the liver, so they need to be taken for life. And the 2 main drugs class of drugs that we have are these nucleoside analogs that work on the VAL enzyme that is necessary for the virus multiplication. And we have interferon alpha and its derivatives that work on the stimulation of the immune response of the infected body. So how can we see the effect of current treatments? Here it's a slide that is that was kindly provided by Thomas Tu and Su Wang. We can see the infection as schematically as a tree. So you would see that in the infected liver, you have the roots of the tree, which represent an important form of the virus, which is called cccDNA. So these are really the roots of the tree. And then we have in the blood HBV DNA, which we can see as a seed for the of the tree, and hepatitis B surface antigen can be represented as the leaves of the tree. With the infection, what we are trying to do with the current treatments is try to reduce the multiplication of the virus. So you've seen this plane, which is the antiviral treatment that will get rid of the seeds, so the HBV DNA. But in fact, in the liver, we will still have the roots, so the cccDNA, and the leaves, so HBs antigen in the serum of the patient. So we don't eradicate the virus from the liver with the current treatment. So we reduce the viral load, but we don't eradicate the virus. So one of the key obstacles to cure HBV is what we call cccDNA, what was represented as the roots of the infection. So cccDNA appears to be long-lived in liver cells. So and it's really called a viral mini chromosome. It's like the chromosome that we have in the cell, but it's a virus, and it is an archive of the virus. So this archive persists in the infected cell, and it is responsible for the persistence of the infection. So the obvious question is, can we get rid of cccDNA, and if so, how can we do it? And so far, nobody knows how to do that. What we know is that in patients who have an acute infection and resolve this acute infection, the vast majority of cccDNA is eliminated by the immune system. But the cccDNA is not always completely eliminated during this resolution of acute infection. And then the immune system can keep this residual cccDNA, so these roots, under control of the immune response. So this is a situation that we observe when someone resolves spontaneously from an acute infection. And the idea and the objective of the treatment is to reproduce in chronically infected people the situation that we observe in the resolution of acute infection. And this is what we call a functional cure. So the functional cure of HBV is defined by undetectable HBV DNA and HBs antigen in serum. Sorry, people are calling, which is not good. So it's the same thing when we don't. So the functional cure is defined by undetectable HBV DNA and HBS in serum 6 months after treatment cessation. So we wouldn't see the leaves and the seeds in the serum after stopping treatment. And this is associated with the lack of disease progression. This may be associated with decreased cccDNA burden in the cell to very low levels, similar to resolution of acute infection. And the immune control, the immune response would control any residual cccDNA in the body. So this is what we call the functional cure. And it is different from a complete cure, which would eliminate all cccDNA. The functional cure does not, and its functional cure is more similar to what we observe in the spontaneous resolution of acute infection. So now in terms of objectives of treatment, what will cure therapies look like? What we know from all the research that has been done in the past years, that we will surely need combination therapy because we'll have to tackle the issue of cccDNA durability, and we need to eat the virus from multiple angles, including the stimulation of immune responses. So the cure therapy will likely be long, around 1 year. So this is much better than lifelong therapy and will require safe drugs. So what types of tools do we have to aim for a cure? So there's a lot of drug discovery efforts that will be discussed later on. And the cure discovery research falls in 3 main categories: drugs that directly act on the virus, drugs that will act on the host cell, so the cells that are infected, to block the infection or to block the replication, the multiplication of the virus. And the third category is drugs that stimulate the immune defense of the body to attack the virus and control infected cells. And there's a lot of work ongoing, and this is really exciting. It is being done in academic sector, universities, in research lab, in the clinic, as well as in biotech companies and big pharma. So this is extremely important. And here is just a kind of a scheme to show you that with all the basic science that is done in research laboratories, we have learned a lot on the mechanism of the virus. And now we have moved from that to the identification of novel targets for treatments, novel biomarkers that are all in clinical studies. You will hear about exciting studies in phase 2 with the aim of improving the cure rate and to prevent hepatocellular carcinoma. Now, how can we cure the infection? There will be different types of approach. Removing the underlying virus multiplication, so trying to remove the seeds, so HBV DNA, suppressing the virus protein production, so this is mainly removing the leaves on the tree, and trying to activate the host immune response to clear the infection and eliminate potentially the roots of the infection. So the new concept for combination therapy are classified in 3 categories: those that inhibit the multiplication of the virus, those that inhibit the viral protein production, and those that stimulate immune responses. And now we are at a stage where all these assets are in clinical development in phase 1b, phase 2 clinical trials. Another real question is, how are we going to combine these strategies, this 3 main class of drugs, in clinical trials, in clinical studies, to really achieve a functional cure, the elimination of HBS in serum, after treatment cessation that should be sustainable after treatment cessation. And we are at this very exciting time where we want to test these different strategies. So this gives a lot of hope for patients, as you can imagine, and for doctors and scientists, because we have now a lot of tools to work with and to do the right and relevant clinical studies to achieve a functional cure rate. We hope in the first stage up to 30–40% and maybe more in the near future. And here I think we, as a kind of perspective, I think with all the research effort done by the community, the entire community, and with all the clinical studies that are being done, I hope we can achieve a cure of the infection in a significant number of patients within the next decade. And then we'll have to work in parallel on the public health issues, and mainly access to care for this disease. And with this, I will stop and leave the floor to the other speakers.
Well, thank you very much. This was an excellent representation of the possibilities to cure hepatitis B. We hope one day we will be able to burn the tree. And this is, I think, an extremely hopeful message for patients and people that live with viral hepatitis B. Moving on to our next speaker, I have the honor to present Dr. George Papatheodoridis, professor in medicine and gastroenterology of National and Kapodistrian University of Athens, and director of academic gastroenterology department and liver transplantation unit of Laiko General Hospital of Athens, Greece. He is also president of the Hellenic Transplant Organization, and he was member of EASL Scientific Committee Governing Board and in EASL and AASLD. He has been also participating in numerous right now organizations and many other initiatives worldwide, but also in Greece. So, Professor Papatheodoridis, the floor is yours. And the presentation has to do with the clinical perspective of hepatitis B cure research. Thank you very much.
Thank you very much, George. I would also like to thank for the invitation to participate in this webinar. Let me just... Okay, yes, I hope you can see my slides. So my task today, following Professor Zoulim's presentation, which covered many issues about HBV cure, I would like to give you my perspective, I would say, as a clinician, why and we need the HBV cure. Do we need it first? And if yes, which we do need it, why do we need it, and which are the anticipated benefits from agents that can offer to a substantial proportion of our chronic hepatitis B patients cure? So Professor Zoulim already mentioned, but this is a chronological map of the advances in HBV treatment. And as we can easily see, most of the agents developed in the previous actual decade, in the decade of 2000, while so we have two groups of agents: interferon, which has antiviral and immunomodulatory activities, but due to several limitations is not used by the vast majority of the patients. And we have the oral antivirals, which are all given as one pill per day. And because of their resistant profile, entecavir and the two tenofavirs, tenofovir disoproxil fumarate and tenofovir alafenamide, are currently used as monotherapy by the vast majority of our patients. And if we use such a monotherapy, what we can achieve, we achieve viral suppression, which means inhibition of HBV replication as it is expressed by serum HBV DNA, which is undetectable in the vast majority of the compliant patients and remains undetectable for many years. We don't have resistant issues with tenofovir, and the rate of resistance is negligible for endecavir monotherapy, except for patients who have developed lamivudine resistance in the past, who cannot be treated and are not treated by endecavir. The drugs are generally very safe. There are some concerns for a small proportion of the patients treated for many years with tenofovir disoproxil fumarate, but this is a small proportion and we have other options. They have histological, these drugs offer histological improvement, so liver histology improves, there's no necroinflammation, fibrosis is reduced, and we can often achieve even a reversion of pre-existing cirrhosis, at least at the histological level. We reduce, although not eliminate, the risk of hepatocellular carcinoma, and overall we improve survival, and the improvement is remarkable, I would say, particularly in patients who do not have cirrhosis before treatment. So we have good treatment options. So why do we need to try for cure? Because without cure, first of all, we have restricted treatment indications. So because we cannot cure all the patients, we don't treat all the patients with chronic HBV infection. We treat only patients with active liver disease. And there are several comments, several recommendations how we can differentiate these patients, the patients who are called to have chronic hepatitis B, the others are considered to have just chronic infection, but some of them have actually very, very high levels of HBV replication. And as I already told you, there's the residual ACC risk. We don't achieve HBV antigen clearance except for a small minority of our patients. And because of that, because we don't eradicate the virus, we have high relapse rates after treatment discontinuation. So most of our patients have to receive treatment for almost for life. Even if we discontinue, treatment is restarted. And in addition to the clinical aspects, there are several impacts on the patient. So particularly the persistent HBs antigen positivity, we know that it may have physical, emotional, professional, social impact on our patient's life, and there is also stress, stigma, and discrimination. So there are several effects of chronic HBV infection in addition to clinical complications. And here is an example of what we can achieve if we achieve even not cure completely, but what we currently call functional cure. So clearance of HBs antigen, the leaves from Professor Zoulim's tree, and you see that if patients achieve HBs antigen clearance, then the cumulative ACC incidence is represented by the line in the bottom of the graph, which is almost negligible. It's much higher in patients who remain HBs antigen positive, but with no HBV replication, at least as we define it by serum HBV DNA undetectability. And of course, the highest ACC rates are observed in patients who not only remain HBs antigen positive, but they maintain some detectable HBs antigen levels. So HBs antigen clearance, even for ACC, is the best endpoint to reduce the incidence. So the anticipated positive effects of at least the functional HBV cure is that we can offer to our patients at least finite treatment duration in substantial proportion of them. We will achieve reduction, I hope almost elimination of the ACC risk, particularly if we achieve HBs antigen loss before the age of 50 and before our patients develop cirrhosis. Hopefully, we will be able to expand the treatment indications. As I told you, they have been rather limited today, and this will have favorable effects of any impact that HB may have on any aspect of the patient's life, on stigma, stress, and discrimination. And if we achieve HBs antigen clearance, if we achieve functional cure, this will hopefully have several positive effects on more important aspects of public health issues like the underdiagnosis. Hopefully there will be more campaigns, the underdiagnosis rates will decrease, so we will be able to treat more patients, and we will also achieve decrease in barriers to treatment, as we already have done with hepatitis C. So I'll stop here and thank you very much.
Thank you very much, Professor Papatheodoridis, for this very interesting presentation. And I'm very honored right now to introduce our next speaker. All these research and all these developments are being done, of course, to serve people that live with hepatitis B around the world. So our next speaker, with a funny last name, which everybody knows as Silvana, Lesidrenska, is the chairperson of HepActive, which is a patient association in Bulgaria, and also a fellow and a board member of Liver Patient International. HepActive was founded in 2009 in Sofia and is a group of patients which main goal is to provide information and raise awareness for viral hepatitis in Bulgaria. HepActive has a wide range of support for people who live with liver diseases, but especially with hepatitis, in testing, consulting, and linkage to care, of course, in raising awareness all over the country. The achievements of this association are numerous. And Mrs. Silvana, the floor is yours to share with us the patient perspective for hepatitis B cures. Thank you.
Hello, everybody, and thank you, George. I hope you can hear me good. I'm a patient who is living with hepatitis B all of my life. It was a vertical transmission. My mom is affected too. So when we created the association 15 years ago, I didn't know anything about hepatitis. So what I'm doing right now is trying to provide the need of support that I was needed 15 years ago. And I will give you only very brief presentation about patient perspective, about treatment, about the disease, about clinical trials. And I hope you see it. So as I said, the patients have many difficulties in life, and they can be from understanding this very serious thing like cccDNA, what is functional cure, what is fibrosis. Patients have to deal with health system. They may fear everyday fear of death. They can face stigma, discrimination. Maybe they live in a country that they don't have access to proper care and treatment. Maybe they have to pay some medical expenses and bills. And it's very hard to combine this chronic disease that you know that you're going to live through your whole life, to combine it with your everyday, with your work, with your family, with your social life. And I think the hardest part is really to understand what does it mean, hepatitis B, because it's extremely complicated, and normal patients could not understand all the so difficult words and medical expression. And it's also very, very hard to have a healthy approach for this disease, and to minimize the negative impact that can have on your daily life. And I think because I contact with patients every day, and I do consultation every day, and I think what is most important for people is, can I transmit the disease to my loved ones? If I'm undetectable, this make me less contagious, or I'm still contagious. How to deal with mentally with this thing, and how to accept it as a normal disease, because usually, especially in my country, for hepatitis B, there is a lot of myths, and people do fear to disclose their disease. So I think patients, we were expecting that we will have the same attention that hepatitis C had the past past 10 years. And I'm really happy that now we are facing this, let's say, rising in the clinical trials and this focus that we see right now. And patients are eager to understand much more about the disease, about what is functional cure. And I know that it's extremely important for a patient to participate in clinical trials, but many times when patient called me and asked, okay, I was proposed by a doctor to participate in clinical trials, but I don't know anything about it. Can you help me? So probably most of you think that for a patient, clinical trials is something like this. Well, thank you for inviting me, but actually it's something like this. A lot of questions that you're popping in your mind. You start thinking, I'm going to take some substance that nobody knows what it is. Is it safe? Am I going to die? What does it mean, functional cure? How to deal with this with my family? How to make the right decision for me, because this affects my health directly. So I think a patient needs more information for sure. And I think it's very important to have easy to understand information about the clinical trial. What does it mean, cccDNA? What is the importance of quantitative S antigen test? What if I participate in clinical trial? What if it's an expected side effect occur? How you will guarantee my privacy, because patients do not want to be disclosed that they've been in clinical trial. And of course, what the speakers before me said, the treatment has to be easy to be taken and stored, has to be finished with no or at least limited side effects, and when it's on the market, should be affordable so every patient could have access to it. And I'm happy that we are here together today, because it's amazing to see, and I'm really touched to see that patients with hepatitis B have their place in the discussion, and it's time to speak more about the disease, to speak more about the clinical trials, about the research. So thank you very much for inviting me. I'm with the time, so.
On time, of course. Thank
Yeah.
you very much, Silvana. Always the patient's perspective is very important when we're talking about treating chronic diseases such as viral hepatitis. I think we need to move to our next speaker, who is Markus Cornberg, who is medical director of the German Liver Foundation and professorship and clinical director of the Helmholtz Center for Infection and Research and director of the Center of the Individualized Infection Medicine. His clinical focus is on the treatment of patients with infectious liver diseases, and Professor Cornberg will have a presentation regarding registries of hepatitis B.
Yes, thanks for the invitation on behalf of the TherVac B group, which is a EU-sponsored consortium which has the aim to develop a therapeutic vaccine for the ultimate goal of to achieve functional cure. And Fabien Zoulim already highlighted the importance of functional cure and what it means, and also George Papatheodoridis showed the impact what it has, and I think also the contribution from the last speakers was very important. Also the patient perspective, what does it mean to participate in clinical trials? And for the clinical trials, I think it is also a bit different than in HBV than in HCV, for example, because in HCV you have such a homogeneous infectious disease. Almost every patient is quite more similar than patients with hepatitis B, where we have the different phases and also different patterns of the viral markers, which is important to consider. So first of all, this is a slide showing just a bit pointed to the, in a nutshell, what TherVac B is a EU program and has the aim, as mentioned, to develop an innovative new therapeutic hepatitis B vaccine to achieve this functional cure in patients with chronic HBV infection. You can see also the web page. There's a lot of information about this consortium, which is The speaker of the consortium is Professor Ulrike Protzer from Munich. And she has developed this idea also in mouse models, there are data that this is an interesting approach to boost the immune response. And Fabien Zoulim pointed this out with this machine that put the flames on the tree to eliminate the cccDNA or the infected hepatocytes by the immune system. Now, let me see. Now, what is the European HBV Registry, which within this consortium or within this project? So we had the idea to establish this registry for patients with chronic HBV infection in order to document the patients before actually they enter into clinical trials. So the aim is to recruit patients with chronic HBV infection who are in principle eligible and willing to participate in clinical trials to cure HBV. This does not mean there's a commitment to consent to a clinical trial. But patients that are interested for clinical trials and to be recruited in this registry before the clinical trial starts, because I think it's very important to monitor biological and host parameters in these patients before actually the study starts. As mentioned, HBV infection is such a heterogeneous disease, and also the patterns change over time. So HBs antigen can spontaneously decline. Also, we know that age is associated with HBs antigen levels. So I think to really have an idea also when we start smaller studies, phase 2 studies, it is so important to have a well-documented patient cohort that enter these trials. Otherwise, we will get wrong information out of the data, and it's I think very important. And also during the registry times, the patient can ask what is available, what are the future, what is the new data. Maybe the patients can get educated from the physicians. I think that has also pro maybe for the patient. And there's no commitment in the end to enter a trial in the end. It's a multi-center study. I will show the centers of the consortium at the end. So why is it good to join? Yeah, you will become a patient at one of the TherVac B study centers that can also inform you about the project, inform you about maybe other clinical trials, about the future of HBV therapies, because all these centers are in the European consortium, are also in EASL members and so on, attend the meetings. I think this is also important. TherVac B will perform routine visits and document your data in the registry. I told you around about the rationale to do this. This helps on the other side selecting patients for participation in specific therapies or clinical trials, but also to document the natural course of the disease before this. And I think this will also help to understand maybe the natural cause of the disease. We will collect also data. We will collect maybe patient samples. We have new technologies also in the centers, omics data and so on. There's a new, also new methods, AI-based method and so on that can be maybe used in these registries to have more important data in chronic HBV infection. Where is the European HBV Registry currently active? We have around 10 centers throughout Europe. At the moment in Italy, Spain, UK, and Germany. We have also one center in Tanzania, which has a different approach and different patients. Also, African patients are part of this consortium. But you can see there are centers in UK, in Germany, and you can see the centers here in Barcelona and also here in Parma, that recruit patients into this registry. Who can participate? So everyone who is an adult chronic HBV patient, more older than or equal than 18 years, up to 70 years, living with chronic HBV infection for at least 1 year, so documented HBs antigen positivity, e-antigen status should be documented for at least 6 months, willing to sign the informed consent, untreated or treated, that doesn't really matter. We have also some exclusion criteria, co-infection, living with clinical relevant concomitant liver diseases, significant comorbidities, receiving immunosuppressive treatment, cirrhosis at the moment we still discuss this exclusion criteria that would also not be, that the patients are in the end also would be not eligible for the clinical studies in the end. That is more or less the rationale for the exclusion criteria. How can I participate? If you're willing to participate and you are especially living close to a center, what I just shown, and you are interested in the TherVac B registry, that could be an opportunity, of course, and you can contact us via this homepage, and then you will be referred to a TherVac B study center closest to your place of residence, and the study physician will then decide together with you and a possible inclusion in the registry, amongst other things, based on the set of clinical criteria. And all patient data, and this is important, very sensitive patient protection, patient data protection, all patient data will be safely stored and carefully handled according to the consent received and full compliance with current data protection regulations. And I think this is very important and sensitive, and therefore it's so important that the patient has also a voice. And I think to understand what is the importance of a study and the clinical trials and so on, I think also this registry could be a very important also tool to educate and to inform patients what is the pro and the con of a clinical studies and what does it mean in the end, the functional cure. With this, I would like to end my short talk. Thank you.
Well, thank you. Thank you very much for this very interesting presentation of such a big and very promising project. We will move now to our next speaker, who is Dr. Pietro Lampertico. Dr. Lampertico is a full professor of gastroenterology, director of the gastroenterology and hepatology division at the Fondazione in Milan, Italy. Professor Lampertico is primarily involved in the clinical management of chronic viral hepatitis at patients, particularly those with chronic hepatitis B and chronic hepatitis D. His research interests includes the treatment of patients with chronic hepatitis cirrhosis due to hepatitis B, the long-term outcome of cirrhotic patients undergoing antiviral treatment, and the diagnosis and management of antiviral resistance to oral nucleosides analogs. Professor, the floor is yours.
Well, thank you very much, George. I hope you can hear me and see my slides. So thank you very much for inviting me and for giving me the opportunity to present very briefly the IPcureB, which is a large European project which has been funded by the H2020 program. Is the immunoprofiling to guide host-directed intervention to cure HBV infection. And the overall aim of the study, I think, is very clear and is perfectly aligned with what has been said by the previous speakers. So the overall aim is to develop novel HBV cure strategies with finite, which means short-term duration of treatment, to improve the management of patients, to prevent the complications, and to expand the number of, increase the number of patients that will be treated. And to achieve this, we are going to develop or implement a cutting-edge program to develop novel immunotherapeutic approaches to cure chronic HBV. It is great, it's my great pleasure to present all the participant centers in the large multi-center European study, which is led by Fabien Zoulim, who is the chairman of this program. As you see, many countries are involved, of course, also the ELPA association. And really the overall idea was to combine different expertise in different fields of hepatitis B, from patients association to expert in cost-effective analysis to clinical studies or clinical centers, to the best of very good immunologists, virologists, and biologists, for example. And the overall strategy of the trial of the project has been or is illustrated in this slide. From now we are basically suppressing viral replication by oral therapy, but the future, as indicated by a previous speaker, is the complete cure or functional cure, and to go from just viral suppression to complete cure, I think we have to integrate clinical activities and non-clinical activities. And really, this is really the HBcureB project, which is based on a proof of concept study or clinical trial. I will show you this data very rapidly, but is also based on many other activities, not strictly or directly related to the clinical trial, which include immunological activities, virological activities, even in vitro or in small animal experiments. The overall idea is to increase the functional cure rates by using innovation, by using innovative immunological approaches to identify the best combination regimen and to end up with large clinical trial, really leading to optimize cure rates. This is the core of the project in the clinical study, which is a phase 2 randomized European multi-center open label exploratory study, and which is will include 100 patients fully suppressed e-antigen negative or nuke therapy for at least 3 years and negative for DNA. Patients will be randomized to either continue oral therapy up to week 76 or to continue oral therapy for week 28 and then stop therapy, or add on toll-like receptor 8 agonist for 24 weeks and then stop both drugs for up to week 76. And the overall idea is to, let's say, stimulate the immune system by 2 strategies. One, to stop oral therapy, this is arm B, or to stop oral therapy after combination with this toll-like receptor 8 agonist, which is oral therapy. To make a long story very short, is like an interferon, but we are not giving interferon, which is injection with many side effects. We are giving an oral pill, which is much better tolerated compared to interferon. So all the advantages of interferon, potentially without the disadvantages. And to organize this study, we have predefined the governance of this clinical study, including a trial steering committee, independent data monitoring committee, and including several sites in France, in Germany, in Italy, and in Spain. The primary objective of the study is to perform a proof of concept study in value suppressed patients. And the primary endpoint is S antigen decline above 1 log. Is not functional cure, we agree, but the duration of the study is rather short. And if you achieve a more than 1 log decline, you are very much committed probably to achieve functional cure, with a long list of secondary endpoints, efficacy and safety endpoints, but also many exploratory endpoints. And I think one of the major plus or the most important advantage of the study is that during the clinical study, many biological samples will be collected, not only plasma, not only serum samples, but also FNA. And this is a new procedure of a liver biopsy, or really limited invasiveness for a liver biopsy, providing material for immunological studies. This is something new and potentially very important. The overall idea, again, is a clinical study, but which leads to, provides material for clinical virology, PBMCs, liver material, which will be used by all the other work packages in the study to really provide a comprehensive virological, biological, peripheral and intrahepatic and immunological studies. We are in track with this study. Many activities have been already performed, and the first participant to the clinical study will enter in the clinical study by the end of this year. And we are in track to end of the study and the clinical trial by the end of 2024. Thank you very much for your attention.
Thank you very much for this very interesting presentation. We will move forward to our next speaker, Camila Picchio, public health PhD research in IS Global, University of Barcelona, currently focusing on health system response, specifically to viral hepatitis. Her research focuses on simplifying the diagnosis of hepatitis B virus and hepatitis C virus using novel diagnostic tools and linking patients to care in Barcelona and sub-Saharan Africa. She is the project manager of hepatitis B Comsava study in Barcelona, screening West African migrants for hepatitis B and linking patients to care or vaccination. She is also working on integrated care in Tanzania and Uganda as project coordinator of the European Commission funded Intel Africa project for the work package IS Global leads. Of course, will be a very interesting presentation because due to, of course, huge movements of the populations worldwide and the fact that globalization has made movements much easier, much faster. We experience also in Europe, but also in other countries around the world, this changes between populations. So we're very looking forward for this presentation.
Thank you very much. Thank you very much for the introduction and to the organizing committee for the invitation to present our project, which is based in Barcelona, Spain, which is titled HBV Comsava. I have to click. Yes. Okay, so I'm going to be focusing on this point of view, which actually Dr. Zoulim showed in his last image in his PowerPoint presentation of this hike going up to the cure, and on the corner there he had public health and access to care. So I'm going to be presenting HBV Comsava, which focuses on this aspect. The main objective of HBV Comsava is basically to link patients to care. Of course, we have some objectives, but really our ultimate goal is to reach this population and get them engaged in care, which we consider, in the case that they're positive, linkage to specialist care, or in the case that they're negative and qualify or are not already have previous immunity, to vaccination. So in order to do this, we use different tools. Namely, we use, this is a community-based intervention, which I'll get to in the next slide, but these are the tools that we use in this project to really to achieve this goal, which is to link patients to care. So we use rapid test and the plasma separation card, which is similar to a dried blood spot sample. However, the final outcome, instead of being a dried whole blood spot, it's actually a dried plasma spot. We use the Hepatitis tool, which is this image down in the lower bottom right of your screen, which is actually an interactive learning tool, which was co-created by migrant populations in Barcelona to increase hepatitis B knowledge. And it's available in many different languages, many different migrant languages, which are spoken in the region. Of course, we also have partnerships with the Hospital Clinic and by the Bron Hospital in the city to refer our patients. And of course, this wouldn't be possible without the support of the health department and the communities. I think I should have here a picture as well of Omar and Daniel, who are community coordinators who are part of the migrant populations and really are essential to the team. And lastly, I'll just mention that we also facilitate CAT salute cards, which would be the equivalent to the public health system health insurance. So our intervention or our program is divided into 2 visits, which are already predetermined. So our community coordinators work with community organizations, which can be faith-based organizations like a mosque or a church, or they can be regular community centers for these migrant populations. And we already established 2 visits before really we show up on our first visit. And during this first visit, we do an information session and use the Hepatitis tool. We, of course, obtain informed consent and do the epidemiological questionnaire. And if anyone doesn't have access to the public health system of the region of Catalonia, at this point in time, we also process CAT salute cards. So irrespective of immigration status in Catalonia, we are including we're including everyone. So, and we're also facilitating access to the public health system. So at this point in time, if anyone requests it, we also we start the paperwork to get them these cards without proof of residency or really any other documentation. And we do the rapid test and collect a blood sample. Everyone is getting a blood sample irrespective of the rapid test result. And for those who are positive on the rapid test, we provide an immediate referral to one of the two collaborating hospitals, which are Hospital Clínica and Vaidebron. Then on the second visit, so on the second visit, which we already informed the people who are participating during the first visit when we will return. So it's in the same community center, really. We don't ask anyone to have to travel outside of their community. And we communicate the results for those people who were negative on the rapid test. We provide post-test counseling, or the first dose of the hepatitis B vaccine is offered on the spot. And for the subsequent doses, so the second and the third, we do send referrals to their primary care centers to get those second and third doses. And of course, we provide information throughout. So between the first and the second visit, the samples that were collected during the first visit are sent to the lab, which are done at Vaidebron lab here in Barcelona as well. And so for those people who were negative, we look at anti-core, and for those people who were positive, we look at HBV DNA and anti-delta. In between these two visits, it's been an average of about 23 days between the people who are really screened and either and linked to care ultimately. So this can be what one intervention looks like. We travel to different community centers. This is a women's association, a Senegalese women's association in the outskirts of Barcelona. So just to give you an overview, so far actually it's been about exactly a year since we started the project, and we've screened about 400 patients, or 400 people, of which the majority were male. The majority came from Ghana, so they're of Ghanaian origin, about 42 years old, and 28% had recently arrived. So we consider recently arrived people who had arrived within the last 5 years to Spain. And the overall prevalence so far is 8.6%. So like I mentioned, we screened about 400 people. And however, this data is, I take this with a grain of salt because the data is always changing. This is an active, ongoing project. So this weekend we have more interventions and these numbers will change. Important to note the surface antigen, so those who were positive, who were linked to care, we have documented first visits for 16 people. However, the remaining actually chose not to go to one of the two participating hospitals because it was too far away for them. So these people actually chose to go to their own primary care center or go through the standard referral process, and we don't actually have documented first visits of them. However, they have reported that they have gone, but we have no way of knowing this. So this is something that we're working on strategies really to improve and get more hospitals on board. Okay, so the HBV Comsava has shown to be a positive or an effective strategy to link patients to care. However, like I mentioned, I think more can be done to increase those who are positive and get them really to a hospital close to them. We're also able to vaccinate. So this is an effective strategy to increase vaccination among this population, which is a high-risk population, coming from highly endemic countries. And the use of simplified diagnostic tools really facilitates this ability to travel into the community and test in decentralized settings, getting patients to care sooner and in comfortable settings. However, more work is still needed to increase the linkage to care percentage. Overall, globally, we have about 75% of people are returning to the second visit. And I showed the breakdown of those numbers kind of by category. But of course, we're still missing 1 in 4. So there's plenty of work to do still. And thank you.
Well, thank you very much. This was a very interesting presentation. Fantastic work that you're doing over there. I think I will need to pass the floor now to the host of the event, Capucine, for there's a bunch of Q&As that already have been answered, but I think there are a few of them that we didn't manage to. I think we will need to address them to our speakers. So Capucine.
Hey, thank you, George. Thank you, everybody. Thanks to the speakers and for the questions too. So a few of them have been answered, but I think many of them were revolving around the participation of patients to the trials that are underway. So I think that was addressed by the two main consortium, but I was wondering whether maybe Silvana has further questions around that and impact she wants to discuss on how to participate in trials in Europe. There's a question around the participation of Eastern Europe, which is a high prevalence region. So is there any possibility, is there any trials happening in Eastern Europe, for instance, not necessarily on these two consortium, of course, but there's any expansion being thought in these regions? And I can direct that either to Markus or Fabien or Pietro.
I mean, if I can start, I'm not sure if you know, some companies will run trials also in Eastern Europe. I'm sure they will, and I could only encourage them to do so. But this is all heavily discussed also in the ISL governing board when I was there, and I'm now out. But to really include Eastern Europe more in the community also for research and studies is a very important issue. And this is, I think, and I tried during my time at the governing board to do things that Eastern Europe will be more and more involved here. We had relationships, for example, with Poland a lot. Yeah, one of my postdocs, Jerzy Rosewicz, did some work on HBV, and he's doing this in Poland with Robert Flisiak and so on. I can only speak now for Poland, but I think this is very important because the prevalence is high in Eastern Europe.
Thank you, Markus. And about that, kind of continuing on that, for the moment, do you have any data on who has been registering to your registry in terms of, for instance, origin and gender, or is it not yet available?
What do you mean, restriction in the?
Well, which population have been registering to your registry, to the patient registry? Do you have any data already on?
No, not yet at this stage.
Okay.
Only the number of patients so far, not in the hundreds. So we cannot say a lot of things. Of course, the heterogeneity of HBV, as mentioned, is high. And for example, in Germany, we have, of course, more patients from Turkey and Eastern Europe, also former. So in Russian and so on, immigrants, and in Spain they have, in Italy it's different, of course. So that will be, of course, interesting to see this heterogeneity on the different populations in the end. Yeah.
Great.
If I can go back to the previous question, the answer is yes. There are new companies developing new drugs for hepatitis B, already working with East Europe. I do not know exactly where or which are the centers being involved, but the answer is yes, because of all the considerations already made. Many patients are very high clinical expertise and so on and so on.
Thank you. And would you be able to know which companies, even though not which centers?
Eventually, yes, but yes, probably yes. But if there are some, websites like clinical.gov where you can find everything, not only the studies, but also the countries where the studies are really run. So.
Thank you. Sounds good. We'll try to gather that information for the future for the ICE-HBV website, so it's easy to find for people living with hepatitis B. Thank you, Pietro.
And I think it's that's Fabien here. Just to add on it, I mean, it's something that we are obviously, as clinicians, we are obviously aware of, and we try to do our best to enroll all possible patients, but then we have to face reality. So when you are in one, I mean, when you are, especially when you are running investigator-initiated studies, it's extremely difficult to organize and manage international studies, even within Europe, because you have such a complexity for clinical research, so many regulatory issues, that it's extremely difficult to run international studies. For pharma industry, it's a bit different because they have obviously more resources. I mean, we are as independent investigators, academic investigators, we have public resources that are much more limited than the industry, and they have specific CROs to take care of all the regulatory issues that is extremely difficult to deal with, and I'm sure Pietro and Markus can comment on that. But so it's reason why, I mean, we are I think a little bit behind the issues of covering, having a coverage of the whole geographic components. So that's so we would need to work on that, I think, and to have patients' voice to speak to, for instance, here we are in Europe, to the EU, Commission, and say, hey, we should help the investigators and help patients to run trials across borders within EU.
Yeah, if I can make a comment, I agree with Fabien. I think that's a crucial point. We are facing this problem every day, and even with the IPcureB project. And to my knowledge, there will be a change in the EU rules in such a way that probably in the near future would be much easier to generate all the documents and we will not have probably to submit the protocols to 25 different ethical committees in different countries, but probably only one. So things will change, hopefully, and we're really looking forward for these changes.
May I add something very quick?
Go ahead.
Yeah.
Actually, in Bulgaria, we have clinical trials for Hep B. And what I have observed is that doctors are struggling to find new patients. Even if it's a small clinical trial, it's very hard to find like 10 or 15 patients. So I think it would be very helpful. I'm just proposing to work in closer collaboration with NGOs, because patients organization actually knows those patients who might need or who might would participate in clinical trials. So in the past 2 years, I was approached by many patients who wanted to participate in clinical trials, and they've had talk with the doctors, and still they have some doubts. I'm not allowed personally to to push them or to say do not do it. It's their choice. But I think patients need more clear information why we need clinical trials. And also we have to have in mind that with this COVID panic right now, a lot of people still think that vaccines and clinical trials, they will take your DNA and blah, blah. So we have to be very clear on that the clinical trial is, the main purpose is to help patients in long term. So I really think that collaboration between medical society and patient association will be really crucial for us. Yeah.
Thank you, Silvana. Markus, do you want to have a comment?
Excellent, Silvana. This is exactly what we need. I think also to educate and the community about the need of clinical trials. And you see this now with COVID, all the people that do not want the vaccine because they fear that there's some microchips installed in the vaccine. I think this is so important. This is for us, it's this is completely clear, but sometimes not for the patients, and you have communication barriers, and I think we need to work on this. Thanks for this. And I need to go now, unfortunately, so goodbye to everyone, all the best. I have another meeting, so sorry for leaving. Bye-bye.
Thank you for joining, Markus. Thank you very much. We're just talking with, so Fabien was mentioning, and Pietro, the policy questions around regulation in the EU. And I know that the two Georges have been involved into a Hep B and C policy association, the EU level. So what do you think we can do there? at the EU level? How do we think we influence the at the European Union level? Do we influence regulators to make these trials easier?
I can start. I don't think this is an issue for hepatitis B. I think this is an issue for any field in medicine, clinical medicine, particularly where multi-center trials are being conducted, because it's meaningless to have so many approvals or applications for approvals for the same trial, and sometimes to have, hopefully not often, but sometimes, occasionally, you may have even different decisions. So the rules should be the same throughout the European countries, at least, because we are considered to be actually one big country, and one central approval should be enough. Because actually each hospital, I will take as an example my hospital, very often do not have the expertise, do not have the people who will be suitable to really look into the trial protocol, understand the trial protocol, understand the limitations. So they just superficially, I'm not sure even if they read the whole protocol. So we are going through a process of just bureaucracy, just to maintain some regulations. But I don't think this is a field that, I mean, we can push and discuss, but this is something that should be an initiative from all fields in clinical medicine. And I was not aware of what Pietro said, and I'm very glad if at some time point this could be achieved. So we will save some approvals.
Yeah, I think you're right, George. I mean, the first comment that you made is a right one. This is, I mean, this is independent of the nature of the disease, and it applies to all type of clinical trials in all medical fields. So I think here. I don't know if there's a way to have patients' voice. So all patients' association would have to speak the same voice to the EU Parliament, because then this is very strong. Then this is becoming very strong, because this is the patients, so the ones that are in need of new treatments, of innovation, and across disease. So it wouldn't be a competition whether is it cancer more more important than diabetes and hepatitis and whatever. This is all. I mean, the issue is the same for everyone, and this was a very important point that you made. But I think, but I don't know the EU Parliament at all. So maybe I think that Philippa is Matthews is on the chat. So I don't know if she can join. If she could speak, because that could be very interesting to see how we can have how can we put pressure on the EU Parliament and make the regulation changing. I mean, that's otherwise we can speak for years with good.
We lost connection.
Yes. Are you okay to say a few words? Sorry, put you on the spot like that.
No, it's great. Thank you for the opportunity to speak. Sorry, I don't have a camera, so you can just see my little icon there. Thanks. I work on hepatitis.
Goodwill from patients. Goodwill from.
Oh.
Connections coming in and out, isn't it?
Exactly. I think now it's fine.
Yeah. So I work on hepatitis B in the UK. I'm a clinician and researcher, and very much interested in this field. So we've gathered some an interdisciplinary group of clinicians, researchers, nurses, academics, and patients, and then connected with the World Hepatitis Alliance, Hepatitis B Foundation, and the British Liver Trust to really try and get some momentum behind this idea about better patient representation, and really interested in lots of ways that we can use that to promote activity, which includes advocacy, education, political lobbying, just visibility of Hep B in the public space. And also, very importantly, as has been really nicely represented, I think the need for patients to be provided with opportunities to participate in research, but actually also involved as active participants in how trials are designed, what are the important questions, how does participation in trials actually affect people, what do participants want out of clinical trials. So there's a real kind of engagement right from the grassroots of how studies are set up and designed and represented and made accessible. So we're really at the early stages of, we've just submitted an abstract to EASL, which may or may not fly, but kind of representing our group and making this point. And likewise, we're writing a perspective piece. But I think this is really important in terms of kind of unifying voices from across different settings. And I'm very keen to link up and hear from others and think about how we work on this together, because it's not something that we can do in isolated silos. It's got to be linked up, and obviously lots of people thinking very much along the same lines.
Thank you.
Thank you, Philippe. I don't know, Fabien, if you want to comment around that. And I think that's something that also at ICE-HBV we can start thinking about a bit more closely.
Yes, sure. ICE-HBV would be a good thing, and I think also all the European associations. I mean, here we are talking about liver and hepatitis B, but so EASL, and the European Liver Association, the different ones, should get together and try, because everyone, I mean, each of these associations or consortium and so on have their contacts at the EU level. So we, at some point, we need to speak one voice and go all together.
Thank you. And I think this kind of events is helpful to build a voice. So if there is any comments from George to close the session.
Well, thank you very much, Capucine. It was an interesting meeting, and it was very nice to hear that in the end of it, we had, specific goals for the next ones, for the future steps. We spoke about clinical trials, about all these possibilities of expanding and finding a possible cure for hepatitis C. Really, a really interesting meeting. I really enjoyed it, and thank you very much for inviting me.
Thank you, George. Of course, everybody, please email us if you have specific question on cure research and even just hepatitis B in general. As we build this frequently asked question for you, it's really useful if we receive your questions. So please contact us anytime, and we'll consult you when we build a FAQ, of course. So thanks, everybody. Thank you to the speakers and panelists. And also feel free to send us suggestions for topics for the future and the information that you need from us. Have a good day.
Bye-bye.